Hello, good afternoon. Welcome back to our conference. We will be starting the pitch presentations, which will be followed by the final keynote session of the day. So I would just like to remember everyone that you can post all the questions you have on the YouTube streaming, which is in the IPB's YouTube page. Please post your questions. And for the people asking where they can find the posters are all displayed in the website of the conference, which is hosted in the Escola Superior Agraria's webpage. So please go there and you will find the conference posters. So without further ado, we will go on to the first pitch presentation, which will be given by Andreia Quaresma, which is a member of the Mountain Research Center. At the end of all the pitch presentations, we will have a short moment of discussion. I would ask everyone that will be presenting to keep their time to the strict necessary. And Andreia, please, you may start.
Good afternoon. My name is Andreia Quaresma, and my work consists on the identification of the origin of collected pollen. This is one of the pea products that have long been used in humans' diet and which is recognized as beneficial for human health. The data that I'm presenting today were obtained in the framework of Insignia, an European citizen scientist project. 108 pollen samples were collected from pollen traps set up in front of beehives from 10 European countries in an attempt to capture a wide botanical diversity. The samples were then sent for our lab in SIM for metabarcoding analysis. But before any analysis, the samples were homogenized in order to obtain homogeneous subsamples and split into two sets. One set was sent to the Institute of Apiculture in Germany to be analyzed by palynological experts, and the other set was analyzed for metabarcoding in collaboration with CBU. The goal of these two analyses was to assess the reliability of metabarcoding before its implementation on Insignia project. To that end, relative abundance at family level were calculated for both methods and compared. Overall, palynology and DNA metabarcoding show a good correlation, with Greece showing the worst results. Some outliers can be spotted, which are being reanalyzed to make sure it wasn't a sequence artifact. Furthermore, the four most abundant families were found for each country, and Rosaceae, Fabaceae, Asteraceae, and Brassicaceae are the preferred plants by bees. From 10 countries, 11 families were found as most abundant, suggesting that bees are foraging in a wide range of plant taxa. Although these preliminary results suggest that ITS2 metabarcoding is a reliable alternative to the classical palynology, we are Sanger sequencing bee plants to enrich the ITS2 database in the hopes of improving the taxonomic classification. The second year of Insignia is underway, and the samples collected are being prepared and analyzed. I would like to thank the Insignia Consortium, CIMO, the Portuguese beekeepers that providing us pollen, and the CBU team. Thank you all for listening.
Thank you very much, Andreia, for a nice presentation, for keeping to the time. So we will go to the next presenter, which is Bianca Rodrigues de Albuquerque, who comes from Brazil. Bianca, whenever you want, please.
Hello, everyone. I'm Bianca. I told you will share with you part of my work, flavonoid composition in vitro antiproliferative activity of the hydroethanolic extract of Garcinia mangostana pericarp. Garcinia mangostana, also known as mangosteen, is a tropical fruit native from South Asia, but also found in other tropical countries, such as Brazil and Colombia. This fruit is very appreciated for the pleasant taste of this pulp, but the consumption and the process of the mangosteen fruit can generate a high amount of bioresidues, signs that more than 6% of this fruit correspond to an edible dark purple pericarp. So, in order to propose a valorizing of this bioresidue, this work aimed to evaluate part of the chemical composition of the mangosteen pericarp, namely the flavonoid compounds, including anthocyanins, and evaluate the antiproliferative activity of its hydroethanolic extract. For it, the extract was obtained for maceration with ethanol and the water, and the flavonoid compounds were identified and quantified by HPLC system with mass spectrometry detector, and the antiproliferative activity was evaluated by superrhodomine BSA used for human thermocell lines. So, as a result, we can see that mangosteen pericarp extract shows a total of 11 flavonoid compounds, begin of them 9 no-anthocyanin flavonoids and 2 anthocyanins, and prostaglandin compounds were the most abundant compounds flavonoids found in this bioresidue. In total, the extract showed 53 milligrams of no-anthocyanin flavonoids and 3.66 milligrams of anthocyanins per gram of the dry extract. And regarding the antiproliferative activity, we can see that mangosteen pericarp extract has a high antiproliferative activity, signs that low concentrations of the extract were necessary to inhibit 50% of the growth of all tumor cell lines evaluated. So you can conclude that mangosteen pericarp could be an interesting source of the bioactivity compounds, and after more studies, these compounds could be applied in several industrial fields, including pharmaceuticals and food industries, for the production of the nutraceuticals and the functional foods. Thank you very much.
Thank you, Bianca. Once again, very strict on time. And once again, I will try and tell everyone once again that we will have the questions at the end. So now we will go on to our good friend Francesco Di Gioia, who is Italian, but is working now at Pennsylvania State University. So Francesco, please.
Thank you, Marcio. Thanks for the invitation to present this with my colleagues Spiridon Petropoulos and Erin Roscoff. We prepared a very short presentation to talk about tailoring food crops to address specific dietary needs. And we want to highlight the case of microgreens, which are considered nutrient dense immature greens harvested after a very short crop cycle at the cotyledon stage. And the idea that we want to present is that there is the opportunity to basically tailor the mineral profile of microgreens using different agronomic biofortification techniques. And here I would like to present what we did in a couple of studies, one in which we were looking at enriching 3 different species of brassica microgreens with iron and zinc. And this is the experimental setup replicated 3 times, and we used basically zinc sulfate as a source of fertilizer for the zinc. And with this experiment, we showed that it's possible to increase from 75 to up to almost 3 times the content of zinc compared to the standard greens that were not fertilized with zinc. And similar results were obtained using iron sulfate. And we see in this case that the response is really species dependent. So the selection of the species that are more suitable for this purpose, for enriching the content of iron or zinc, is very important. Beside enriching nutrients, we also have the opportunity to reduce the content of specific minerals. And here I'm reporting the case of low potassium microgreens that were studied as a potential opportunity to address the issues of patients affected by hyperkalemia, which is basically toxicity caused by an excess of potassium in patients that are affected by chronic kidney disease. And this was again the experimental setup, and we used 3 levels of potassium: the standard rate, which is 100% of the reference level, and then 25% of that level and no potassium. And this was the outcome, and we showed that it's possible to reduce from 50 to 80% the potassium content. And this was obtained without reduction of yield. And also there was an effect a little bit on the shoot size, but also we saw enhanced total phenolic content and antioxidant compounds. With this, I would like to thank our sources of funding and the people that help us with this project. And thank you again.
Thank you very much, Francesco, for that nice presentation on microgreens. So we will go on to our fourth presentation pitch. It will be given by Isabela Ribeiro, which is also a Portuguese researcher. Isabel, whenever you want, please.
Okay. Okay, so good afternoon, everyone. My presentation will be on exploring cranberry extract ketones and films towards the improvement of antimicrobial food packaging. Okay. It is a fact that foodborne diseases are a problem worldwide, and also that there is an emergent need to reduce plastic consumption and find more sustainable food packaging. So new strategies to prevent food waste and deterioration and the food packaging system are a need. The aim of this work was the development of ketones films incorporated with cranberry extracts to produce new biodegradable, eco-friendly films with enhanced antioxidant, antimicrobial, and anti-biofilm properties. Before films development, we had to characterize the cranberry extract activity and composition. So we observed that the films had antimicrobial activity towards the tested bacteria and antioxidant activity. Different phenolic compounds were identified through LC-MS analysis, and also it was possible to detect A-type procyanidin compound, which is an important compound regarding its antibiofilm activity. Knowing that our extract was suitable, we proceeded with the production of films, and we produced 2 ketones films, one added of PEG as plasticizer, and the other added of PEG and glycerol as plasticizers. Films produced retain a good light protection, especially under UV light. Water contact angle measurements we observed a low contact angle, which can be an advantage considering that several bacteria tend to adhere more to hydrophobic surfaces. Water vapor permeation and oxygen permeation were suitable. And regarding the mechanical properties of films, films added of PEG presented a higher extension percentage. Regarding the film's antibiofilm activity, we had a very good antibiofilm activity with this cranberry extract films. No SCR was detected on the surface. And regarding E. coli, few bacteria were observed. and some of them were dead, which are here stained in red with the Propidium iodide stain. As a conclusion, we can say that it was possible to identify phenolic and other compounds responsible for the antimicrobial and antioxidant activity of these films. The properties evaluated assured film suitability for food quality maintenance. Films also showed antioxidant and antimicrobial activity, and a significantly reduction on E. coli and S. aureus biofilms was an outstanding outcome. Finally, I would like to thank my colleagues from the Faculty of Pharmacy and my other co-workers from the Nova University, and also the audience for their attention. Thank you.
Thank you very much, Isabel, for the nice presentation. So we will go to the second to last presentation, the pitch presentation, which is another researcher from the house, José Pinela. José, you're ready, please.
Good afternoon, everyone. I will present some results of an extensive study that aims the valorization of Solanaceae crop byproducts as a source of bioactive ingredients. Tomato is highly consumed worldwide, and its annual production generates a considerable amount of plant byproducts. Although tomatoes are well known for their composition in nutrients and bioactive compounds, little information is available in literature regarding tomato leaves. Therefore, considering the massive production of these biomass and the current trends of circularity and sustainable development, this study was performed to evaluate the phenolic composition and the bioactive properties of tomato leaves provided by the Portuguese Gene Bank located in Braga, Portugal. So the samples consisting of pruning materials and end-of-cycle plants were used to prepare hydroethanolic extracts, which were used to characterize the phenolic composition by HPLC-dead ECMS and used to evaluate antioxidant activity by the DPPH and reducing power assays, the anti-hemolytic activity using sheep erythrocytes, and the antimicrobial activity was tested against foodborne microorganisms by the serial microdilution method and the colorimetric assay. The chromatographic analysis allow identifying phenolic acids and flavonoids, as we can see in the bar chart, with prevalence of quercetin 3O rutinoside or rutin. We also found that the pruning material had a higher polyphenols content than the end-of-cycle plants. The leaf extracts show antioxidant activity with a better performance than the corresponding tomato fruits previously characterized by us. Some pruning materials were also effective in protecting the erythrocytes from the oxidative hemolysis, with results close to that of Trolox, the positive control. And some extracts also had the ability to inhibit and kill some bacteria more effectively than the antibiotic ampicillin. Therefore, we conclude that table tomato crop byproducts can be recycled to produce rutin-rich bioactive extracts for possible use in the agri-food sector as natural preservatives. Finally, I want to thank my work colleagues, the supporting entities, the conference organizers, and everyone for listening. Thank you.
Thank you very much, José, for the nice presentation. And now we will go to the final presenter of the pitch sessions. I will try and pronounce your name correctly. So it's Seymanur Ertosun, from Turkey. Please.
Can you hear me well?
Perfectly.
Okay. Good afternoon, ladies and gentlemen. I will be presenting you the nutritional, technological, and sensory attributes of bread enriched with bee pollen and bee bread. When bees visit the plants, they collect the plant pollens and they mix with their secretion enzymes and the nectar. And this mixture agglutinate, and this is how bees upgrade the basic basic plant pollen to bee pollen, and some of these bee pollen kept in the hives, and bees cover on top of it, and this is this closed environment lead to anaerobic lactic acid fermentation. After that fermentation process, we have new product called bee bread. But how did we use these products? We basically substitute the total flour amount with these two bee products separately. We prepared 3 different ratios of enriched breads, and along with them, we also prepared conventional bread in order to compare in terms of nutrition, phenolics and flavonoids, antioxidants, and bread properties. When it comes to nutritional analysis, we are seeing that when we compare with the conventional bread, there's an increase in terms of protein, fibers, and energy, and this increase quite promising. When it comes to specific volume, which is really important for the consumer's choice, there's not much of a negative impact of our bee products over the conventional bread. And when it comes to the color analysis, the more we increase the ratio, the color change is more visible. And when it comes to total phenolics and flavonoids, also with the antioxidant capacity, we are seeing and observing the same pattern is followed, which means that when we increase the ratio, we are also increasing the antioxidant capacity of our enriched bread, and the conventional bread has the lowest antioxidant capacity. And for the texture profile, it's really important for the consumer's choice in the market. There's not much of a followed pattern in this texture profile. We can say that there's not much of a negative impact over samples, over bee products, over conventional breads. And finally, for the sensory evaluation, we tested with 15 participants, and they more likely to prefer the bee bread over control and also bee pollen. But for both of the samples, our participants more likely to prefer the enriched breads when they compared with the conventional bread. Thank you for your time.
Thank you very much, Seymanur. So we are running a bit late, so I think we've got time for a couple of questions before we go on to the keynote preferred by Luis. So I've got a question here for the first presenter, for Andreia. I think she's still there. Do you think the metabar coding can be used as a complementary tool, or is it essential to distinguish the origin of pollen?
Metabar coding is now being optimized to replace completely palynology, because palynology needs a lot of expertise, and with metabar coding, we can also add a little bit deeper of taxonomic classification and go to the species. So we want to replace palynology.
Thank you. Thank you very much. For Francesco, do you think what would be the biggest difference between this reduction of potassium between doing it in microgreens or in normal greens? Is there a difference in their the way of doing?
Yes. So that's a good question. There are 2 reasons at least to consider microgreens instead of standard vegetable. One is the fact that because of the short cycle, we can really use no potassium and still have something good to eat. The other component that is important is that microgreens are considered nutrient dense and are rich of vitamins. And the people affected by this issue normally have to boil vegetable to eat them. And then they lose all the vitamins. So this will be a very good way to still have a taste of vegetable without renouncing to vitamins.
Exactly. Thank you very much, Francesco. I have a question from my colleague here, Dr. Sandrina Heleno. She's asking if you, Isabel, sorry, I think I said Isabel. So if you analyze the stability of these biofilms over time, or was it just stopped in time?
We didn't observe through time. We had a 24 hours experiment, give 24 hours for the biofilm production. And after that time, we analyzed the produced biofilm on samples.
Thank you. Thank you very much, Isabel.
Okay.